Hello guys. So today we're going to understand the process of osmosis. But before we do, here's some retrieval practice. Pause the video now and have a go at those questions. So, have we done? Great. Let's have a look at some answers. So, what type of organisms are bacteria? Bacteria are prokaryotes. If you got that right, well done. Prokaryotes don't have a nucleus. So, in bacteria, the DNA is free inside the cytoplasm. Question 2. Which gives a higher resolution, light or electron microscopes? Electron microscopes have a much higher resolution. Cell differentiation is when cells become specialised. Before they are specialised, they're called stem cells and they can change into lots of different types of cell. But once they differentiate, they become specialised and exist as one cell type. Question 4. The sperm cell has got an acrosome, which is in its head, and it helps to break down the egg cell so that the sperm can join with the egg. It's got lots of mitochondria to give it energy to help it swim, and it's got a tail to help it move in a particular direction. Chromosomes are strands of DNA that contain genes. We have 46 chromosomes or 23 pairs in a human. Mitosis is used for growth and repair. Question 7. Diffusion is the movement of particles from a high concentration to a low concentration until we reach equilibrium, where all of the particles are equally distributed around the space. 8. 2 examples where diffusion may take place. Now, you've probably come up with lots of different ones, but the 2 I came up with are alveoli in the lungs and the small intestine during digestion. Question 9. Give 2 substances that can diffuse through cell membranes. Well, I've given you a few more there, but we've got oxygen and carbon dioxide. They're both really small and can pass through the cell membrane where other larger particles or molecules, like in question 10, can't pass through. So fats, starch and proteins are all too big to pass through the cell membrane. So today, our success criteria, we're going to look to define the term osmosis, predict what happens to plant and animal cells when placed in different solutions, explain the behaviour, and evaluate why plant and animal cells behave differently. But before we do that, have a go at this. Pause the video now and see if you can recall some information about diffusion. All done? Great. Let's have a look and see how you got on. Here's the mark scheme. So diffusion of nutrients happens across the wall of the small intestine. To increase the efficiency of this, the walls are covered in villi. The villi give this small intestine a large surface area. The villi walls are only 1 cell thick to decrease the distance the nutrients need to cover to get into the blood. They're also covered in capillaries, which increases blood flow to the area, meaning that the concentration gradient is always maintained as the nutrients are being swept away from the area as soon as they've diffused. So let's have a look at where we'll get marks then. So to increase the efficiency, the walls are covered in villi. That's going to be your first mark using the keywords. Then the use of villi gives the small intestine a large surface area, will get you a second mark. The fact that the villi walls are only 1 cell thick will get you your third mark, and it helps the nutrients to get into the blood. They're also covered in capillaries, which increases the blood flow to the area. That's going to be your fourth mark. The area maintains the concentration gradient would be 5 marks, and for correct spell and punctuation grammar you will get a full 6 marks for that question. So there's a video clip here that will tell you all about osmosis. Take note of that and you can watch that in your own time, but it tells us what osmosis is, why it's a passive process, and the direction of the concentration gradient, all of which are really important in osmosis. So, hopefully you've had a chance to watch that video. If you haven't, pause this now and go to that YouTube link. Okay, so what's the definition of osmosis then? Hopefully you've got something along the lines of osmosis is the movement of water from an area of high concentration to an area of low concentration across a partially permeable membrane. If you said semi-permeable, that's also the same as partially. That will get you up to 3 marks in an exam, but sometimes it might only be 2. And then, if you can, please make a note of the differences between osmosis and diffusion, because this does come up quite often in exam questions. So, here's your answers. Osmosis is the net movement of water molecules from an area of high concentration to an area of low concentration along the concentration gradient across a partially permeable membrane. And your extensions there too. So, osmosis in cells then happens in both plants and animals, but works differently because they're different structures. So plant cells are able to take in lots and lots and lots of water because they have their tough cell wall which stops the cell from bursting open. Plant and animal cells can both become turgid when they take in lots of water. When plant cells lose lots of water, they become flaccid, and that's why plants can wilt. And when plants wilt, they bend and become quite floppy. And so flaccid is the key term for that happening. If too much water is lost from cells, we say they can become plasmolyzed. If that happens, the cytoplasm pulls away from the cell membrane, but the overall cell stays the same size. But if that cytoplasm shrunk, all of those chemical reactions that go on there won't be able to happen. And there's a little clue here to why this is so dangerous in humans. People who take ecstasy drink too much water over short periods of time, and it's really, really dangerous. Think what might happen to cells in humans. If they take in too much water, they don't have that thick cell wall around them. So, what might happen to the cell? You got it. It bursts. Have you seen these words before? Possibly think about energy drinks, or a person who's described as being hyperactive. They're overactive, right? So hopefully you've heard of hyperactive. Hyper means more active or overactive. Hypo means low or less active. So we can talk about this in terms of sugars and the amount of sugars in a substance. If hypo is low and hyper is high, what do you think iso might be? Yeah, you got it. Iso just means the same or balanced. There we are there. Explains it in a little bit more detail. So if we think about it in cells, then here we've got water moving out of the cell. The cells become plasmolyzed, and the cytoplasm pulls away from the cell wall. In this picture, we've got water moving in and out at the same rate. The cell's nice and healthy. And then lastly, in this one, we've got water moving in. You can see the cell's grown slightly in size. It doesn't actually grow. It just swells up as it takes in water. And so which one is the isotonic, the hypertonic and the hypotonic solutions? What do we think? So in the first one, there's a low water concentration outside the cell, so water moves out. This is a hypertonic solution. In the second one, it's isotonic. We've got equal water concentrations inside and out. And in the last one, we've got a high water concentration outside the cell, so water's moving in by osmosis, and we call this hypertonic. So this is how it might appear in an exam. The diagrams show plant cells. Plant cell A is in a hypertonic solution, and diagram B is in a hypertonic solution. Now the wording is very, very similar, hyper and hypo. Remember, hypo means low, hyper means high. So what's your definition of a hypertonic solution then? Pause the video now and have a go. Okay, great. So a hypertonic solution is one that is more concentrated than the cell or the cytoplasm, and that would get you 2 marks. Well done if you got that right. So here, what word can be used to describe a plant cell placed in a hypertonic solution? Any ideas? Pause the video now and have a go at these questions. So a plant cell in a hypotonic solution, we describe it as being turgid. So it's taken in lots and lots of water because there's more water in the surroundings than there is inside the cell, so the water moves in by osmosis. And a hypertonic solution, if a plant cell is placed in it, then water would move out of the cell and the cell would become plasmolyzed. And lastly, explain what's happening in diagram B. In diagram B, the solution outside contains less water than inside the cell, so the water moves out by osmosis. The cytoplasm pulls away from the cell wall and the cell becomes plasmolyzed. And finally, just some questions for you to think about. And that's it for today, guys. So thanks for listening and well done for taking part. Bye.